Welcome to PPLS Perspectives, a podcast bringing students and academics together to explore the research carried out by the University of Edinburgh School of Philosophy, Psychology and Language Sciences and the impact this work has on society.
Welcome to Season 2 of the PPLS Perspectives podcast, where we talk to academics about work currently being done in the School of Philosophy, Psychology, and Language Sciences here at the University of Edinburgh. My name is Lucas Perry. I'm a PhD student in psychology, studying the genetics of executive function and development. And at time of recording here in Scotland, it happens to be Dyslexia Awareness Week. And in recognition of that, we're going to be talking about dyslexia and its genetic components with Professor Michelle Luciano. Michelle is the Personal Chair of Behavioral Genetics here at the University of Edinburgh. She is a differential psychologist interested in cognitive skills in both childhood and later life, and has a special interest in reading ability and language development. Welcome, Michelle.
Hi, Lucas.
So to start with, can you elevator pitch your research for us? Just give us a quick 2-minute summary of what it is you do.
Sure. So as you mentioned before, Lucas, I'm what's known as a behavioural geneticist. So I'm interested in understanding the genetic and environmental influences on behaviour, particularly human behaviour. And my research has looked at traits like personality and, as you mentioned, cognitive ability. Today, I'd like to focus on my research area of dyslexia and reading skills, and how I've been working with collaborators across the world to better understand the genetic influences and associations with reading skill and reading difficulties.
So maybe a good place to start with this is to talk about what dyslexia really is. Dyslexia does have kind of this lay concept of words getting jumbled up on the page. So maybe you can talk a bit about how you think of dyslexia as a researcher and some misconceptions you think people might have about it.
Yeah, sure. So my view is that dyslexia, which is, like you said, a reading and spelling difficulty that people, and usually it's kids who first get identified as dyslexic because it's noticed within the education system that perhaps their reading isn't as fluent or as accurate as other children. And so with dyslexia, it's not really about jumbling letters. So what kids and adults have difficulty with is decoding words, so being able to sound letters out. So in English, we're typically taught that letters or combinations of letters, which are known as graphemes, are related to a phoneme or a sound. And so if we know these rules, then we can sound out new words that we come across if they're spelt phonetically. Of course, with English, not all words are what is known as regular words, so that we can use these decoding rules to sound them out. So some words we need to remember. We store these in our lexicon. But dyslexics seem to specifically have, or mostly have, problems with this phonological processing route. So they have difficulty with those grapheme phoneme conversion rules and applying them.
So does that then imply that dyslexia is less of a problem in languages with more straightforward spelling?
Yes, that's what we would expect in terms of accuracy, but in languages that have better phoneme to grapheme mapping, people with problems or dyslexics might be slower to read, so they have problems with fluency more so than their accuracy.
So what's your origin story? How did you come to this research?
So when we're thinking specifically of today's research that I'll be sharing with you, it really started out during my PhD when I was looking at twins to dissociate the proportion of genetic and environmental influences on a trait. And during my PhD, I was interested in cognitive abilities more broadly, so including working memory and processing speed and higher or complex cognition that you would measure in IQ tests. And in those days, it was using twins just to separate the influences of nature versus nurture. So it was, as technology progressed in genetics, it came to the point around probably the early 2000s, after the human genome was sequenced in its entirety, that genotyping methods became much cheaper, and one could move to actually locating or identifying specific genes that were associated with particular traits. So I've for a long time been interested broadly in lots of different cognitive abilities, but I reoriented a lot of my effort to focus on reading and spelling skills, which are just so important in terms of progress in education. They're vital skills, and I really wanted to better understand the genetic influences on those particularly, because I think there's potential for impact of genetic research in these areas.
That's a big change, moving from those twin studies to the more molecular genetics methods. Can you talk to us a bit about how you actually do that kind of research today?
Yeah, sure. So one of the studies I really wanted to highlight in today's interview was a big, what we call a genome-wide association study, which we conducted on dyslexia diagnosis. And so when we perform one of these studies, we firstly need to extract DNA from an individual, and usually, or ideally, you would have a blood sample, but you could use saliva samples or buccal swab samples. And then people working in labs will extract the DNA, and what they do, or use machines to do, is genotyping. And so when we talk about genotyping, we're talking about identifying the sequence of nucleotides that make up our DNA. So there's 4 nucleotides of DNA, and there's millions of nucleotides that vary between individuals. So where a nucleotide varies at a single point along our DNA, this is called a single nucleotide polymorphism. So we're able to do genotyping, which will result in potentially millions of single nucleotide polymorphisms, which we'll refer to as SNPs. And then when we've measured a trait on an individual alongside their DNA or their SNPs, we can look statistically at associations between these SNPs with the trait. So the trait that I'm interested in, or for this study that I'll be describing, is dyslexia diagnosis. And so basically we're trying to see whether a particular nucleotide at a SNP is higher or lower in frequency in dyslexia cases versus control, so people who don't have dyslexia. And so then we can, depending on the statistical association results, we can see whether a SNP is associated with our outcome.
So it's millions of nucleotides you're looking at. So it's not just one gene for dyslexia you're trying to find.
That's right. So we look, so in the latest study, and this was the first really well-powered study of dyslexia, we looked at around 8 million SNPs. And some of these SNPs will be located in genes and others won't. And so we don't really understand what their function is. Even those SNPs that might be associated with dyslexia that occur inside a gene, we still don't have a clear idea on what their role is. It might be that they're the causative SNP, but it might be that they're associated with another SNP or variant in the genome that is having the causative effect. So actually changing a protein, for instance, that is affecting whether it's a brain process and then ultimately behaviour.
Okay, so when we say causative SNPs, that doesn't just mean that people who have those SNPs are doomed to have dyslexia and there's nothing that we can do, does it?
No, so in fact our results from this big genome-wide association study, which had over 50,000 dyslexic cases and over 1 million controls, found that actually there were many SNPs associated with dyslexia, but each of them had a very small effect. So there was no SNP individually that was located in a particular gene, for instance, that was increasing the likelihood that someone would be dyslexic or not. So it was lots of SNPs working together, and this is what we call a polygenic effect, when we've got more than one gene influencing a trait. So it's very, in terms of what we understand about the genetic influences on dyslexia, there's lots of them, and our study isn't even able to capture all the genetic variation we know that is associated with dyslexia. So if we look back at family studies, and many of these are twin studies, they show that dyslexia is up to 80% heritable. So that means of the variation in the population with dyslexia, if we look at the genetic versus environmental influences, around 80% of the differences we see between people in dyslexia diagnosis are due to genes. And our genome-wide association study is only capturing about 15% of this heritability. So heritability is the genetic, the proportion of genetic variance in a trait over the entire variance, which would be genetic and environmental variance. So there's still a lot we don't know about the genetic influences on dyslexia, but what makes it even more complicated to understand is that genes and environment can interact. So we don't know whether some of these genetic effects that we see are associated with dyslexia might actually not have an effect if they were being expressed in a different environment. So it's very much probabilistic. So if we knew somebody's genetic propensity to dyslexia, we wouldn't have any certainty that they would actually be dyslexic or not, because of course environment might modify the effect of genetic predisposition.
So what are some of the challenges involved in conducting this kind of work?
Some of the challenges. So we were, in terms of this study I'm discussing, which was the largest study of genome-wide association of dyslexia to date, we were working with a consumer genetics company called 23andMe. So this is a company where you can pay to get your DNA genotyped, and the company can provide you information about your ancestral background and other information about genetic variants you might have that are associated with different outcomes, like whether your hair's curly or not. And these customers of 23andMe can opt in to survey research. So our study was limited, I guess, or one of the challenges of our study is that we had what's known as a selected sample. So the customers of 23andMe weren't representative of the general population. So in fact, if we looked at the dyslexia prevalence within our sample, it was around 5%, whereas in the general population, you would expect it to be closer to 10%. So we had a selection bias in our sample, but because it was so large, we did manage to overcome this somewhat, as we were still successful in finding significant SNPs or associated with dyslexia. Another challenge of this sample is that we had a very big age range. So we were looking at adults who were self-reporting a dyslexia diagnosis. So we had 18-year-olds up to age 110 years. And you can imagine that people 100 years old probably weren't getting identified with dyslexia at school. So we had to check whether older people in the sample, that their results might look, whether their results look different to younger people in the sample who were going through an education system where dyslexia was more routinely identified, whereas with the older participants, there might be participants there who didn't know they were dyslexic. But we were able to overcome that by doing a separate statistical analysis of younger versus older participants, and we could show that the genetic findings were virtually identical, so there were no differences between those age groups.
Very nice. So what's the impact of all this research? If we can identify these genetic variants associated with dyslexia, what can we do with it?
Yeah, so at the moment, like I said, we can't explain the complete heritability of dyslexia using these common single nucleotide polymorphisms that we looked at. So there's no way that we can use our genetic association findings to be able to predict, for instance, kids who are likely to be dyslexic or not. But that would obviously be a really useful thing to be able to do, because we know that with dyslexia, the earlier the intervention, the better chance for success. So if we were able to predict who is at increased likelihood of being dyslexic, by just knowing their genes, which of course don't change during the lifespan. So this is information that we could have from birth if we wanted, and we could then implement very early intervention programs even before kids start school to help support them with reading and spelling. So our study is nowhere near that kind of, we don't have the reliability of genetic prediction yet, but what we can do is use our results to do further research, for instance, in the area of gene by environment interaction. So what many researchers do with their genome-wide association study findings is to calculate what's known as a polygenic index. And this is a single score that is an index of a person's combined genetic, propensity towards a trait. So if we could, we can do this for dyslexia. It's not reliable at the individual prediction level, but it's useful for research where we're looking at group effects, for instance. So we could then use polygenic scores in individuals who have been genotyped and who have been measured on other traits we might be interested in. So if, for instance, we were interested in whether the environment can minimize the genetic kind of propensity a person might have towards reading difficulties, we can design studies where we're incorporating a polygenic score and some environmental moderating variable. It might be related to socioeconomic status, it might be related to how reading is actually taught within schools, whether schools teachers use phonics-based programs. So these are really interesting questions that when we get results for, they might be more broadly applicable in terms of identifying environments that are most conducive for kids who might have a genetic propensity to have difficulties in the future. And there's lots of other really neat things that we can do with our genetic results. So we can look at correlations or associations between dyslexia and other traits of interest. And one of our important findings was that we see quite a substantial overlap in genetic associations for dyslexia and ADHD. And this is something that we that clinicians do see. For instance, we know that dyslexia occurs at a higher rate in kids with ADHD than in the general population, but we don't understand why. So studies like ours are able to look at the shared genetic associations between different traits, and here we have found this moderate genetic correlation or association between ADHD and dyslexia. We don't see the same, for instance, between autism and dyslexia. So lots of interesting results that further research could actually show might be useful for clinical interventions even, or remediation programs.
Thanks, Michelle. I think that's where we'll wrap this up. So thank you everyone for listening. I hope you've all learned something about dyslexia today. Please be sure to check out our previous episodes, and we'll see you next time.
Thank you.